Thank you. Could I have my slide? Great. Well, thanks, Eddie. And thanks also for allowing me to present our work here. It's my first in-person Academy meeting. I'm deeply pleased and honored to be part of this fantastic Academy. Now, I guess that everyone here will agree that the human brain is the most complex, but also the most fascinating organ that exists in nature. Our brain contains 87 billion neurons that have to be born at the right time, then migrate to the right position, and then make the right connections in order to allow us to perform all the cognitive processes that we're capable of doing. Unfortunately, however, this complexity comes at a price. Brain disorders are among the most challenging and most unresolved biomedical problems. It's now thought that about 25% of the entire population of the United States at some point during their life will encounter disorder of the central nervous system. What's even worse is that the treatments of these diseases are very problematic. Most of the drugs that exist go back to principles that were found in the 1950s, and so it is now thought that the research in neurological disorders is among has among the lowest success rates. And there are many reasons for this, but one of the reasons is that many of the drugs fail in the clinical stages. And this is because most of the experiments that are done to develop these drugs are done in laboratory animals, particularly in rodents such as mice and rats. And while those rodent animals are absolutely essential for biomedical research, and while they have taught us some of the biggest secrets of the development of our brain, they have some limitations. And one particularly striking example is shown here. This shows you an MRI scan of a healthy patient and a patient who suffers from a severe neurodevelopmental disorder that's called microcephaly. As you can see, the volume of the patient is severely reduced, and that's because the patient carries a mutation in a gene that's called NDE1. Now, NDE1 is conserved. It exists in yeast, in all animals, and also in humans. But when you make the corresponding mouse knockout, not very much is actually happening, and you do not see the same severe disruption that you actually see in humans. And there are many other examples for this, and I would like to remind everyone that a human fetus, as it develops, generates the number of neurons of an entire mouse brain every 4 hours, and this for 9 months. But there is hope. I think we're living in a time where we're experiencing a complete revolution in biomedical research. And that's actually because a number of new technologies are coming together. The first one is we get more and more complete genome sequences. Soon we'll have millions of them. We can generate pluripotent, naive stem cells from essentially any patients through a method that was developed by Shinya Yamanaka, and that's called reprogramming. We can read and write in these genome sequences, much like in a typewriter, through a methodology that's called the CRISPR-Cas technology, developed by Emmanuel Charpentier and Jennifer Doudna. And finally, we can now use these pluripotent stem cells and the gene-edited cells to grow them into three-dimensional cultures that represent various different types of human organs. And it's actually absolutely amazing that the inventors of many of these technologies are members of this academy. And so my lab has tried to contribute to this by generating a method that we can use to grow in vitro tissues that resemble the most complex organ in our body, which is the human brain. And the method that we developed is called the Cerebral Organoid method, and it's summarized in this video. So we start with pluripotent stem cells, which we dissociate after limited protease digestion. Then we take between 5,000 and 8,000 of those cells. We put them into a 96-well tissue culture dish, and then they sink down. They aggregate together. They form balls of tissues that are called embryoid bodies, and that contain the 3 germ layers. Then we change the media, drive them towards neuroectoderm, the tissue where the brain develops. And then we embed them in a substance that is called matrigel, which is a concentration of extracellular matrix components. Then we culture them, first on suspension culture and then on an orbital shaker. And what will happen inside these matrigel droplets, inside these organoid cultures, is the cells will sort out. They will form what's called a ventricular zone, which is shown in red here. The cells will divide, the neurons will migrate out. They will arrange in a layer-type fashion and form a cortical plate, which is a characteristic for the developing human cortex. And then we end up with structures like this one, where on the right-hand side, so you see cortical tissue in the red. I show you the ventricular zone, which is where the progenitor cells are in green, is the neuron layer. And there's other regions, like the convoluted area that you see at the middle and the bottom part of the image, which is the choroid plexus. That's the area that generates cerebral spinal fluid. So we can recapitulate certain aspects of morphogenesis that happens during human brain development, starting from pluripotent stem cells. This is how these organoids look in real life. They are not as impressive when they are looked at by light microscopy in transparent balls of tissue. Sometimes they become very exciting. They can grow the first stages of a human eye, which is shown in the middle. But when we section them, we find that their 3-dimensional architecture very much resembles what is seen in a human cortex. So here on the top, you see an organoid, and on the bottom, you see an actual human cortex. And at this early stages of organoid development, they look remarkably similar. But most importantly, what we can do is we can use them to recapitulate human disease. So for this, we worked with a patient who suffers from severe microcephaly. This is shown on the left-hand side. You can see that the patient's brain is severely reduced. We generated iPS cells. And on the right-hand side, you see the organoid models. On top is the healthy organoid, which contains a big ventricular zone, many neurons, and at the bottom is the patient's organoid. So we can recapitulate the microcephaly phenotype that is seen in the patient. And we can now use this organoid model to ask, why is this actually happening? And we found that the patient carries a mutation in a gene that is called CDK5-RAP2, which is important for the cells to determine the orientation in which they will divide. And we could attribute the disease to this defect. But most importantly, we could then repair that defect. And on the bottom right, I show you a repaired organoid where you can see that the size of the organoid is almost completely rescued. So we can use cerebral organoids to investigate neurodevelopmental disorders and to unambiguously assign particular genes to them. Now, organoids reached prime time in 2015, when the last time there was a global pandemic in the world caused by the Zika virus, which reached Brazil right at the time of the Olympics. And at the same time, there was an increase in the number, a dramatic increase in the number of microcephaly cases. And work from several labs has then used the Zika virus on these organoids and could show that the Zika virus really can cause microcephaly in organoids. So we can use cerebral organoids to probe the pathogenicity of infectious agents. We can also use them to investigate biological processes that are only happening in the human brain. And we've recently analyzed a disorder that's called Tuberose Sclerosis. It's characterized by tumors, which you see on the left-hand side, and by cortical tubers that you can see on the right-hand side. And we could attribute this disorder to a series of cells that we call the clip cells that exist in the human brain, but they don't exist in the mouse brain. And we could show that these clip cells in tuberous sclerosis patients, they overproliferate, they generate tumors, and then the neurons that are generated from these cells migrate into the cortex, where they give rise to the tubers. Now, why am I telling you this? I'm telling you this because these clip cells have a very important endogenous function. We can actually grow organoids for over a year, and then the neurons that are generated by these cells, they migrate in long streams that you can see on this slide. And through a whole series of experiments, we could show that this is recapitulating a process that is happening in the human brain, but not in the brain of rodents. When a human child is born, the brain is very incomplete. There is a massive migration of neurons into the cortex that is happening after birth. And our data indicate that these are the neurons that are generated by these clip cells. And so now we have a handle to investigate this human-specific process and to ask why is it happening and what is its role. And we can already say that probably this increased complexity arising from this process is what causes our unique susceptibility to tuberose sclerosis. So we can use organoids to analyze various kinds of disorders, but the most important thing is that the function of our brain is actually not just to generate a huge complexity of different cells, but the largest part of our brain is actually connections. And I would like to remind everyone, while the number of neurons in the brain is in the billion range, the number of synaptic connections is larger than the number of stars in the universe. And so where is this field going? We're going towards modeling neuronal activity profiles in organoids. And the reason why we think this makes sense is because organoids do actually generate complex neuronal connections. So on the left-hand side, I show you a calcium imaging experiment. Every time when you see a light flash, the neuron becomes active, and you can see that the neurons are firing these coordinated flashes of activity. And the reason for this is because the neurons arrange in very complex neuronal networks that are shown on the right-hand side of this slide. And we can actually observe how these networks are forming. This is a live imaging movie where you can see the neurons inside a growing organoid and where you can see that they're forming their connections. They have these large growth cones, and they interact with each other and make decisions as to whether to connect or not. And we're very excited to actually observe that the activity patterns of these neurons change in a characteristic manner in patients that suffer from neurodevelopmental disorders. So this is a plot of an electrophysiology experiment from a cerebral organoid. The time goes from left to right, and you can see that about every minute, the organoid is firing a flash of activity. And at the bottom, this activity is transferred to the frequency space, and the activity consists of flashes of high frequency oscillations. Now, why am I explaining this in such a degree of detail? This is because intracranial recordings of patients who suffer from epilepsy have also recorded these high frequency oscillations. And what people have found is that they actually characteristically change in patients, and they characteristically change when the electrodes are placed close to the epileptic core. And this is actually used as a biomarker during surgery on those patients. And so we're very excited that we see also these precise same activity changes, an increase in frequency with which these oscillations are happening in the patients who suffer from tuberose sclerosis, a genetically caused epilepsy. So we can use cerebral organoids to investigate early brain development, to model infectious agents, to investigate human-specific processes, and we are about to establish assay systems to use the organoids to probe neuronal activity changes that are characteristic for particular neurodevelopmental disorders. And of course, these in vitro systems can then be used to test compounds for their activity on these changes, and will hopefully eventually lead to the development of better treatments for some of these disorders. I wouldn't want to end without thanking my entire research group. I think it is very important because this is a very highly dedicated group of excited young researchers. Their names are listed at the bottom. On the right-hand side, I show you the agencies that are funding our work, and I thank you all for listening to me.
Thank you very much, Jürgen, for keeping to time. We have time for maybe 2 questions. Marichuk.
Talk and really enjoyed, and I think the quantifications you made were very interesting. Different than cancer, our knowledge on neurodegenerative disease is much worse. Okay, and I have been on several workshops and discussions that on the days, and you have 2 crowds. You have the people like you're talking about doing imaging, looking at macro things, looking at the organ level or high cellular organizational level. Then you have the molecular people looking at fibrils and aggregates, fiber formation. But somehow, different in cancer, the middle ground, the systems biology gates, where you come with the molecular level and try to figure out what appears to be much more crude on this field. You know why we sort of miss this? You have this sort of 2 communities, but you're missing this link. Connecting the molecular part to the level of science you're doing without the systems biology between, where you look at circuits and pathways and.
Okay, thanks. So I think that's a very interesting question. I think you're alluding to the usefulness of these, in a very general term, to research on neurodegenerative disorders and why, basically asking why the progress is not as it should be. It is a fundamental problem in neurodegenerative disorders. In vitro systems, I mean, these are disorders that are happening after 50 to 70 years, right? And nobody can culture neurons that long. Also, a mouse doesn't live that long. The problem in the mouse is that the entire spectrum, there's no way to recapitulate the entire spectrum of neurodegeneration. So there are Alzheimer's models that recapitulate aspects of Alzheimer or aspects of Parkinson, but the exact same thing just does not exist. Nevertheless, we've learned to accept that, but that leads to certain inconsistencies. In organoids, nobody can grow organoids for that long, and so people have reported abnormalities when they make mutations that would actually cause early onset Alzheimer's, but the data are a bit weak. There is some hope. There is work, I think I can mention it, it's unpublished, but it's from Lawrence Studer, the papers on bioRxiv, that actually has identified a cocktail of chemicals that make cells age faster. And there's great hope in that. And they have also used them on organoids, and the organoids develop certain features of neurodegeneration at a very early stage. So I'm not sure to answer your question precisely, but I try to give you give you my opinion about the confusion in the field. I think the confusion in the field is simply because that there is no perfect model, and for that reason, we have learned to accept inconsistencies, and different people accept different inconsistencies. So the ones who have the mouse models accept that the mice just don't really get the proper neurodegeneration. The people who look at the tau tangles, they accept the fact that it's not 100% for sure that the tau tangles are actually causing the disease.
You talk, no doubt, and I see the potential of making organoids, say, of the liver, or I don't know what, the brain. The brain, I mean, that's the human itself. Have you ever thought about any ethical implication? I'm sure you have, and don't get me wrong, I'm not saying one should not do this, but the brain is something special.
So believe me, I've thought a lot about the ethical implications of this. I'm a member of the board that has written the ISSCR guidelines for working with organoids. I'm a member of the committee of the German Academy of Sciences that is currently preparing a white paper to advise the government on regulations. I think we are far too early to think about ethical implications. What I would like to make a number of points here. I hope I didn't give the impression that the organoid models in any way recapitulate a fully functional brain. I've just discussed with Stanislas a lot about what consciousness means. Consciousness involves the entire brain. I think everyone agrees. Every neuroscientist agrees on this, right? An organoid doesn't do that, right? And it also requires a specific architecture, which organoids don't have. The point that I'm trying to make is they don't have to. They can be very, very useful without that. We do not need an in vitro brain. I'm not sure they will ever help us to understand intelligence. There, I expect more insight from AI and from trying to model it. But diseases like autism or epilepsy or even schizophrenia, I predict that they will help us a lot to understand. And by the way, the liver organoids really don't look like liver.
I will reserve the final comment for myself. I had a student called Yoshiki Sasai who invented the organoids by making an eye in a mouse from stem cells. And he sent me his paper saying, "Dear Eddie, this is my greatest work." And I answered back very courteously, but I answered back, said, "Well, but we have been doing this in frogs since the 1930s. And because they make brains, and if you let them alone, they develop, they make an eye connected to a brain and so on." And I did not see the immense potential that this had, of coming from a stem cell like you have shown now. So it's a great advance for medicine, no doubt. And Yamanaka is also in our academy. And Yoshiki Sasai is deceased. But, yeah.
I think there's many people who have contributed to this, and as we discussed before, the ability to generate three-dimensional cultures in vitro it actually goes way back, right? The Matrigel, for example, goes back to the 1980s, right? And many of the principles have been developed at that time. But it is very clear that Hans Clevers, Yoshiki Sasai have been the pioneers of this. What I think also made a very strong difference in this field was that somebody showed that you can actually apply this for a disease research. So I was very serious when I thought that it's the combination of these technologies that allow us to address human genetics, And it's very, very sad that Yoshiki is no longer among us. I'm sure it would have been absolutely fantastic to have these discussions with him.